we're increasing the carbon dioxide in the atmosphere, and that's what this red line on the graph on the right is showing us. And when the carbon dioxide enters the ocean, a series of chemical reactions take place, and ultimately the pH of seawater goes down. This decrease in pH, or acidification, can create conditions corrosive enough to dissolve calcium carbonate minerals. Okay, it looks like it's going a little slow to advance, so I'll make sure that it's working here. So just a refresher on pH scale. 7 is neutral. That's where distilled water is, and anything below 7 is acidic, and anything above 7 is basic. And seawater is about 8.1. So the pH scale is on a log scale, so even a slight change in the hydrogen ion concentration can have a big change in, even a small change in the pH value is a big change in the hydrogen ion concentration. So here we have the atmospheric CO2 in red again on this graph, and now we're adding the seawater carbon dioxide, the seawater CO2 in green, and you can see that that's also going up through time, and we have the pH in blue, so this is going down through time. So this is showing the ocean acidification trend at the Station Aloha site near Hawaii. This is the Hawaiian Ocean Time Series. So the values are specific to that site, but the trend is seen globally. This is a graphic from my colleague Li-Ching Zhang, and it shows the historic and the predicted surface seawater pH. So we're starting way back in the 1700s, and we're coming up into 2100. Now I want to talk a little bit more about, so we're talking about carbon dioxide and pH on a global scale, but I want to talk about why we specifically want to monitor for ocean acidification in Alaska. So Alaska's waters are naturally more acidic. This is a map by Takahashi, and this is data through 2005. And this is really when folks were really starting to release a lot of information about ocean acidification. And he showed that waters around Alaska, and especially in the Bering Sea, had some of the lowest pH values that they observed. So Alaska's waters are naturally more acidic because cold water holds more carbon dioxide, more gases, just like you think about like a soda in your refrigerator would hold more carbonation than sitting on your counter. And then there's other physical processes at play, like where we sit on the global ocean conveyor belt, and we also have a lot of freshwater inputs along our coast. So things like enhanced sea ice melt or glacial runoff and respiration of organic matter in highly productive areas such as those around Alaska can exacerbate the carbon dioxide driven ocean acidification in high latitude regions. So the scientific community early on identified Alaska as a region vulnerable to ocean acidification, and we opened the Ocean Acidification Research Center here at UAF. This is the Trothuda campus on Tanana Dene land. And we began starting to monitor around the state in 2008. So now, today, there's more than 24 researchers monitoring ocean acidification around Alaska. It's not just me and it's not my lab. This is definitely a community and network effort. So I'm a chemical oceanographer by training, and I'm mostly going to talk about the chemistry. But first, I really just want to talk about the heart of the matter. We care about chemistry because we care about how that affects what lives and grows in the ocean. So the Alaska Ocean Acidification Network is a great resource to learn about species-specific responses, and I'm going to try to highlight a few of those here. But again, I'm not a biologist, so this is not my work. This is the work of my colleagues. So first, I'm showing a scanning electron microscope image of a clamshell. This is from the Kelly lab here at UAF. and they're studying the effects of ocean acidification on several different clam species. So if you go to the network site, there'll be links to resources on different bivalves. None of them are from Alaska. So the Kelly Lab is currently doing the research on the Alaska specific species, but I just wanted to give you a couple of examples of what folks are studying from clams and other bivalves from other regions. There's the impacts are varied. Some there is no difference, some had negative effects. And my point here is that it's important to study a wide variety of species because there can be a lot of variation. So here I'm showing Chris Long. He works at the NOAA Kodiak Lab, and they work on all of the commercial fishery crab species in Alaska. And the take home here is, again, we have varying results depending on the species. The juvenile and adult here I'm referring to is for golden and red king crab. They show negative effects in the juvenile stage, but the adults might be a little bit more resilient. And blue king crab may have an evolutionary advantage. Snow crabs are a different story. So you might have seen this article come out last month about the resiliency of snow crabs to ocean acidification. So this is quite different from king crabs. So you can't just study one species and have kind of a blanket assumption about how they might react to changing ocean chemistry. So snow crabs face a lot of challenges, but maybe ocean acidification isn't something we have to worry about right now. So when I first started working on ocean acidification, I don't think I saw one talk in like 5 years that didn't show a bunch of photographs of pteropod shells dissolving. So this was explained as the canary of the ocean when talking about ocean acidification. Nina Bednarzik is the leading researcher who works on pteropods, and she has worked in the Gulf of Alaska. And the thing I want to highlight here is not only the direct impacts, which we might think of as a dissolving shell on a carbonate species, but also the indirect impacts. So pteropods are a main food source for salmon. So when I think about pteropods, I think about salmon. Fish do not have a calcium carbonate shell, so the impacts are going to be coming from different directions. Ocean acidification can impact the food chain of the fish. It can affect its prey, like pteropods, but it can also affect the fish directly through sensory impacts. So the person that works on finfish in Alaska is Tom Hurst. He works for NOAA Alaska Fisheries Science Center at the Newport Lab. He's mostly looking at groundfish. He's studied a lot of walleye, pollock, cod, some different soles. And here I'm giving an example of his cod work. So he's showing that you might not only have a change in growth or survival, but you might also have an impact on behavior. They found evidence of impacting the way that cod reacts to light under acidified conditions. So the thing I hope you took notice of with some of these examples is it was not all doom and gloom. First of all, there's evidence of resiliency. Juveniles grown under harsh conditions may be able to catch up later. And we're not talking about imminent death for all of these species. So the majority of the research shows that ocean acidification is often sublethal, but also keep in mind that the marine ecosystem is going through a lot of change, and the biggest one is a change in temperature. So this was demonstrated with the snow crab example. The crash is likely driven by increased temperatures. So many of these species might be stressed from multiple variables. That can include warming, ocean acidification, and deoxygenation. So it's the cascading effects of all of these stressors that might bring species to different thresholds. Okay, so I'm at the Ocean Acidification Research Center, and my primary job is to monitor Alaskan waters. I'm trying to figure out the intensity, duration, and extent of ocean acidification around the state. So I have some various groups I'm going to walk through here with a couple of monitoring examples and the ways that different groups will use the data. So I already showed an example of research scientists looking at species response, and those are my colleagues. And now I'm going to talk about some of my own data, and that's collecting water samples and doing remote sensors. Okay, so what are we monitoring? We're always monitoring the physical oceanography. That's the temperature, salinity, and depth. And then we're also monitoring the 4 main parameters of the carbonate system. So that's dissolved inorganic carbon, or DIC, total alkalinity, or TA, the partial pressure of carbon dioxide, and pH. If we measure 2 of these 4 carbonate parameters, we can calculate the other 2, so we don't have to always measure all 4. And we often express our data in terms of saturation states, or omega. This parameter outlined in yellow is always calculated from whichever input pair we choose. So what do I mean by saturation states? Here we're talking about the ability of an organism, a shelled organism like a clam, to either easily pull calcium and carbonate ions out of the seawater to build its shell, or if the water wants to pull those ions away from the shell and dissolve them into the seawater. So if the water is saturated, it favors shell building, and if it is undersaturated, it favors dissolving. So dissolving is a little bit of a hyperbolic word. We see pitting, as shown by the examples from the pteropods and the clams. But again, this doesn't mean that these organisms are dead. It does mean that these shellfish need to use a lot of energy to maintain their shells. And if they're putting energy into their shell, that might be taking energy away from things like growth and reproduction. So we're calculating the saturation states predominantly for 2 calcium carbonate minerals, that's aragonite and calcite. So they have the same chemical formula, they're just different mineral structures. So this is sort of like graphite and diamond, like they're all carbon, but they act and react very differently to different conditions. So I'm mostly going to talk about saturation states with respect to the mineral aragonite. Okay, so we're ready to monitor, and we want to get really good spatial coverage. And one way to do that is to use research vessels and do hydrographic surveys. We work all around the state. This map is showing our CTD stations, where we're profiling with a conductivity, temperature, and depth instrument that has a water sampling rosette. So these sites have data from sea surface to sea floor. We bring the water samples back to the lab here in Fairbanks, and we analyze them for the carbonate parameters. So I'm going to show you a couple examples from the cruise work that we've done in the last 15 years. Of course, I'm going to start in Prince William Sound. So you all might have heard about this paper. It's a little old by now. But Wiley Evans led the analysis and results for this project. And here I'm highlighting the Icy Bay Station. All of our sites are from the western Prince William Sound, and we did a survey there from 2008 to 2017. Here I'm only showing the data from 2011, that's in black, and 2012, that's in red. So the y-axis is the water depth. So this Icy Bay Station is 150 meters deep, and I'm showing you the seawater temperature, the seawater salinity, and this omega saturation state. So the dashed line at omega equals 1 is just a reminder that this is where calcium carbonate minerals want to dissolve. And I'm showing that we have values at this site that are either very close to this threshold or below it for the saturation state for the aragonite mineral, and we also have low values below 1.5 for saturation of the calcite mineral, which is much more durable. So this is a pretty corrosive site. So what are we talking about when we're talking about corrosive waters and ocean acidification in Icy Bay? So first, again, ocean acidification is from anthropogenic CO2 released to the atmosphere that's absorbed by the ocean. So the ocean globally is absorbing this carbon dioxide, so we have that going on. The water here is cold, so it can hold a lot of carbon dioxide, so we have that going on. And we're also right next to a glacier. So this is giving us a lot of fresh water that has a unique chemical signature. And all of this contributes to the corrosivity that we're seeing in the water at this site. So Alaska has many factors making it vulnerable to ocean acidification. So this is an example of a project that we did in the Bering Sea. So this work was written up by Jessica Cross, and we surveyed this area from 2008 to 2010 in spring, summer, and fall. We've continued the survey up until now, but mostly we are only surveying in one area of this map, and we are not surveying very often in the summer, but this is an ongoing project. But the thing I really want to just show you here is we're also seeing corrosive waters with respect to aragonite and calcite in the Bering Sea. So this map is showing us the spring, summer, and fall omega aragonite on the first three panels, and the far right panel with the most red is showing us omega C. And we still see some values, that's the cool colors, the blue colors under 1, that are corrosive even to the more durable form of calcium carbonate. So we have corrosive waters in the Gulf of Alaska, we have corrosive waters in the Bering Sea, and we also have corrosive waters in the Arctic. So this is a project in the Chukchi and Beaufort Seas, and it's called the Distributed Biological Observatory. This is an ongoing project. We started this monitoring in 2017. We had a ship go out this year. I'm running the samples right now in the lab. And the purpose of this project is to be a change detection array to monitor everything from physics to whales in these biological hotspots. So on the map, you're going to see a red box around a transect with the number 3 on it, and that's where these profile data are from. This is transect 3, which is just north of Bering Strait. So on each profile that we have 2017 to 2021 data from, on the right side of the profile is Point Hope, and on the left side of the profile is the Russian-US border. And again, on the y-axis, we have water depth. It's shallow here. It's about 50 meters deep. And we have corrosive waters in a few of these years. So the corrosive waters, again, are below 1, and they're the cool colors. So one thing that's kind of weird about this project, it's kind of good and kind of weird, is that the ship goes out at a completely different time every year. So we're not comparing the same season in all of these years. But what it does tell us is how things change throughout the seasons. So some years we're out there and it's still boom town. It's phytoplankton blooms and zooplankton blooms and everything is growing and living, huge biology going on. Other years we're there after the bloom. In some years, we've even been out in late November when sea ice is forming, and we have formation of cold and deep water, which is setting up a completely different physical environment. But the take home here is that we are seeing corrosive waters in the Chukchi and Beaufort Seas as well. And then just to highlight again, there's a lot of other components to this project. There's dozens of people working on huge amounts of taxa in this region, and they're constantly pulling up lots of gooey grab bags is what I call it. There's lots of life in this area, and once we wrap up the chemistry, we'll be able to give this to our partners to say what conditions are the various biological species growing in, and that might be important to all different kinds of groups. Okay, so next we're going to take all of these data that we have from around the state, and we're going to try to predict the future. So right now I'm going to give you some predictions for the Pacific Arctic region, and after my next example I'll show you some predictions for the Gulf of Alaska. So here I'm showing the average aragonite saturation on the y-axis, and then time on the x-axis, and we're seeing a decrease, which we would expect with ocean acidification through time as we continue to release CO2 to the atmosphere. But right now we're seeing some corrosive waters around Alaska, and in the future we're predicting all of the waters around Alaska to be corrosive. Okay, so we used ships to go out and do spatial surveys, and I was mostly talking about aragonite, and now highlighted with the yellow line, I'm going to be talking about carbon dioxide and seawater. So one way that we monitor this is through surface buoys, and we have two long-term stations. One is in the Gulf of Alaska and one is in the Bering Sea. And we chose these stations because they're the two longest records that we have in Alaska. They're both multi-decadal, going back about 30 years or so. So we know a lot of information about these sites. And we added the CO2 measurements in 2011. So here's the data from the Gulf of Alaska buoy. This is at the mouth of Resurrection Bay and Sunny Cove. This graph is showing us the partial pressure of carbon dioxide on the y-axis, and it's showing us the months of the year on the x-axis. And we have a pretty long record for these types of measurements of Alaska. I'm showing all the different years here with the current raw values in blue. These are just what I'm getting from the daily satellite transmissions. None of this is processed, so it might change a little bit. But the few things that I want you to notice is that there's a predictable seasonal trend. First, the ocean and the air are in near equilibrium in the winter months. The air is shown by the black dashed line around 420. And then they gradually decrease in the spring during the phytoplankton bloom and return back to near air values at the end of the calendar year. So we had a little hiccup in the data. There's no blue dots around May. We had our sensors go offline, and I went down to Seward and fixed them. So we can't see the minimum CO2 from this year, but what I'm hoping you can sort of see is that not only the timing of the spring bloom is different every year, but the intensity is different also. Some years are the drawdown is not quite so strong. So you might not get below 200, where in other years you get really close to 100. So when we're talking about carbon dioxide, we're really interested in the movement of carbon dioxide between the ocean and the atmosphere. That's called flux. And one of the big questions is whether or not different parts of the ocean are a sink or a source of CO2. So if the air is around 420 and everything at that site is lower than that, we would call this a carbon dioxide sink. And this is well known. We've documented through other research that on average, the Gulf of Alaska is a CO2 sink. And globally, the ocean is a CO2 sink, but there are changes in that, and we want to have a good idea of how things are variable around the globe to help us with our big global models, which I'll give an example for later. First, I want to show you M2. So this is in the Bering Sea. This is a seasonal site. We have sea ice here, so we can't have our surface buoy out year-round. So here the months are a little bit different on the x-axis. But the trend is the same. We mostly have values below air, We have a strong biologically driven phytoplankton bloom drawdown. These are plants, right? So plants are taking CO2 up to photosynthesize. They're using the CO2 out of the water. And then through the season, it returns back to air values and mixes in the fall storms. So just like the Gulf of Alaska site, we'll say that M2 is a CO2 sink. Well, except when it isn't. So in 2021, we had a canceled cruise and we went out to go pick up this buoy and we couldn't. So then like chaos ensued and we tried to charter vessels and everything just kept getting pushed back. And the buoy was out there much, much longer than it normally is. When we started getting these fall and winter storms coming in, they have a huge mixing effect. The Bering Sea is very productive. So everything that was growing in the Bering Sea is now dying, falling through the water column and decomposing, giving off respiration signals, adding CO2 back to the water. These storms are also bringing in deep water from off the shelf that naturally have high CO2. And now we're seeing these really high CO2 values at the surface at M2. And this is a temporary CO2 source. So we knew that the Bering Sea outgassed in the winter, but there's very, very few ships and researchers out there at the time to make these measurements. So this was pretty exciting to be able to measure this and get the intensity of the outgassing event. What was not exciting is when a sea ice flow broke off from the main pack and ran over our buoy and ripped the tower off and all the instruments and effectively turned our buoy into a fur seal couch. So this was the guy that was on the buoy when we went out there and picked it up. Okay, so M2 and Gakoa are our long-term sites, and we also had a couple of buoys out for a few years around Port Conclusion in Southeast Alaska, just off Chatham Strait, and also in Kodiak in Chiniak Bay. So I'm going to show you those data. So here I'm plotting air carbon dioxide in blue. This is around 400. This is old data. This is 2013 to 2016. So it's a little bit lower than what we're seeing today in 2023. The red line is the Gakoa buoy that's in the northern Gulf of Alaska. The gray line is Kodiak, that's in the western Gulf of Alaska. And the black line is from southeast, that's in the eastern Gulf of Alaska. So what I'm trying to convince you is that the surface ocean carbon dynamics in the Gulf of Alaska really vary on location. So at the southeast site, we have not only our highest surface seawater carbon dioxide at over 600 microatmospheres, but we also saw the shortest duration of surface water CO2 below air. So Gakoa is a sink most of the year. It has this longest green line when seawater CO2 is lower than air. Kodiak is next. And then southeast has the shortest green line here, which is the approximate length of time that seawater CO2 is below air at this site. So earlier I talked a little bit about CO2 flux, but what else do we learn from carbon dioxide in water? So remember, CO2 leads to this series of chemical reactions which decreases pH and it also decreases the carbonate concentration, leading us to look at, these are showing when it's a source and when it's a sink. And then here we're able to calculate the saturation states based on the carbon dioxide values at these locations. So I have Gakoa here on the left, Kodiak is in the middle, and southeast is on the right. So again, we're looking at saturation states. For aragonite, these colors are opposite of what I was showing before. I apologize for that. This was generated by Wiley Evans and is a great way to look at how the less favorable conditions are shown throughout the year and also how they'll change through time. So the cool colors are the high omega, which are the more favorable growing conditions according to our biology friends. And these mostly occur in the summer months. And through time, the more favorable conditions, the blue is, shrinking, it's disappearing. We're seeing a lot more of these grays. This is the most corrosive water and the least favorable growing conditions. We also see the most corrosive water earliest and for the longest time of the year at the southeast site. So this is showing us that the southeast region is more vulnerable to ocean acidification than the northern Gulf of Alaska. Okay, so the surface observations from the buoys are monitoring CO2, and we can do all of these other calculations to look at things like saturation state. We also want to look at things like time of emergence. So this is the number of years of observations necessary to detect an anthropogenic trend. So that's that human-caused change in seawater pH. So when I was looking at those graphs, you would see all these squiggly noodle lines, and they were always changing depending on the year. Some you had a really strong drawdown in April. Sometimes it didn't happen until May. We have huge variability in the coastal ocean, and that's globally. The open ocean has less variability, and it's the amount of variability that tells us how long it's going to take us to be able to see a trend outside of that. So the open ocean, these years that I'm showing on this map are much sooner than the coastal regions. So on the right graph, we're seeing the North Pacific, and if you look way up at the top, that's the Kakoa buoy, and it shows 2027. So this is when we're expecting to see the anthropogenic trend outside of all of that natural variability that we have. So this is a long game. We need to monitor for a long time, and we need to make really accurate measurements to be able to detect this trend. Okay, so that's me monitoring the water, and I'm not the only person that monitors the water. Marine farmers and hatcheries are also monitoring. So I'm going to show you a couple examples of that work. The first hatchery that we started working at was Aluktik Pride. It's now named the Aluktik Pride Marine Institute. We installed equipment there in 2014 and began tracking the carbonate parameters on their seawater line. This was an awesome experience. We have a really great relationship with them, and Aluktik Pride Marine Institute leads the entire show now. Ocean Acidification Research Center does not. So they are doing all of their own monitoring themselves, and they even started a community sampling project. So stay tuned for results from that project in the future. That's going to be a really exciting summary. I think in the next maybe year or two, we'll see that. So we moved our equipment out of Aluktik Pride when they bought all of their own sensors when they wanted to lead the monitoring, and we moved it to the Oceans Alaska site in Ketchikan. They've had some major infrastructure changes in the last couple of years, so our continuous record is a little bit spotty, but we hope to be able to get this going in the future. But this is really exciting because we want more measurements in Southeast for sure. And the most recent shoreside station that we've partnered with is the Kodiak Lab. They started monitoring in 2019. They're also working on a community sampling project through the Kodiak Area Native Association. And there's one more shoreside station that my lab does not participate on any of the equipment doing the monitoring, but we are partnering through some other projects in Southeast, and there'll be a summary for all of the shoreside stations and the community sampling in the next year or two. So keep looking at that Alaska Ocean Acidification Network site to see the results of that in the future. Okay, so now we're going to move on to the resource managers on this groups, looking at this kind of the research. So I talked a little bit about saturation states. I talked a little bit about carbon dioxide. Now this variable outlined in yellow is pH. So I'm going to show a couple of examples of some pH data that we have. So when we're talking about resource managers, the first stakeholder that's using all of the monitoring that I do is a modeler. So here's an example of a modeler. This is Darren Pilcher. He works at the University of Washington. There's a Bering 10K model. That's a biophysical regional model for the Bering Sea, and it's been adopted by managers and stakeholders to understand ecosystem trends. So Darren adds the carbonate chemistry package to the Bering 10K model. And he's able to create these spatial maps which can help refine regional risk and hazard exposure, especially in data poor areas. It's really hard for us to get out into the Bering Sea all year round, so the model gives us a lot more information. So here I'm showing Darren's modeled observations spatially on the map, and then in the circles is the validated model. That's what we're collecting on the ship using seawater samples. And so these seawater samples validate the model, and Darren's able to make all of the calculations to make everything line up. Models are always a little bit of a stretch, but this one's doing a pretty good job. So depending on which fishery the managers are studying, they might want a different index. So right here I'm showing you pH. So higher pH, 8.1, is the blue colors. You see that in the inner shelf. Lower pH is the red colors. You see that more on the outer shelf. But fisheries managers also like saturation states. So remember from the species examples, we're talking about shellfish, we're talking about finfish, we're talking about all these different things. And depending on what kind of effects and what species we're looking at, you might want to use pH or you might want to use aragonite saturation state. So the pH is to look at the effects on finfish, and the aragonite saturation state is to look at the effects on shellfish. So here Darren's plotting the percent of bottom water against years, because this is a model. He first is doing the hindcast, and then he'll do a future forward-looking cast for two indexes that the biologists set for us. So they're looking at unfavorable conditions at the gray line for saturation states, and then unfavorable pH conditions in the black line. So the amount of bottom water, the percent of bottom water, is increasing with time, and that's a result of ocean acidification. So we have the model validated with the observations. We have the indexes set for the specific fisheries, and then we want to communicate this information through different products. The biggest being the annual ecosystem status reports. There's several. Here I'm showing an example of the Bering Sea, but there's also one for the Gulf of Alaska and the Aleutian Islands. And the point we made in 2022 was that we saw an expansion of corrosive bottom waters, but there was no evidence that that was driving the snow and red king crab crashes in the Bering Sea. So this is a way that our observations can be communicated to resource managers. So the value chain of this data is I do the monitoring, and then Darren puts it into his model with specific index requests from the fisheries biologists. It's put into the ecosystem status report, and this is given to the North Pacific Fishery Management Council, and then this is also given to all the other stakeholders, coastal communities, tribes, everyone else. I do not need to tell anyone in this room that the impacts of changing marine ecosystem can have on a community. You all are the experts on that, so you all can share that with me next. Okay, so one of the big things that's really a part of our mission is to make sure that all of our data is public. So I'm at the base of this pyramid. I'm making the observations, and then I pass the data along up through the pyramid to make sure it's included in the data, in the global products. So right now we're in the United Nations Ocean Decade, and one of the main priorities of this effort is to research ocean acidification and how changes in the ocean carbon cycle are affecting marine ecosystems. So 80% of the carbon is stored in the ocean. This is a huge amount. So all of our data ultimately winds up in the global carbon budget. And check out these numbers. So we have 37,000 gigatons, that's a lot, of dissolved inorganic carbon in seawater. So this is an order of magnitude bigger than any other number you see on this figure. So the amount of carbon that is in the ocean is huge. The last part of my carbon dioxide story I want to talk about is marine carbon dioxide removal. Many believe that just reducing carbon dioxide emissions won't be enough to prevent further warming, ocean acidification, everything else that's coming along with that. So some groups also want to remove the carbon dioxide that's already been emitted to the atmosphere. So this is a developing field with a lot of new research, and a new project was just announced in the last month, including $25 million in monitoring, reporting, and verification of these different types of marine carbon dioxide removal techniques. So the table here on the right is from NOAA's framework that they released this year. So this is just their opinion. There's lots of different opinions out there. I'm not saying any of this is right, but I think it's just interesting. So you'll probably read about a lot of these approaches in the news. And the one thing I want you to take away from this table is that the scale can be very, very different. So the lowest scale potential is on this table is macroalgal cultivation. So what is macroalgal cultivation? That's kelp farms. So this is telling me that there's very low scale potential, and the duration of the CO2 storage is also very low. So this is telling me that kelp is not going to save the planet with respect to mitigating the carbon dioxide. What kelp does is it helps lessen the effects of ocean acidification locally and on very short time scales. So when you talk about this type of effort, it is something in the immediate distance and the immediate future. The highest scale potential on this table is the alkalinity enhancement option that's at the top. And here you have a moderate to high scale potential and a high, very high, greater than 20,000 years for duration of storage. So this is very, very different than what we're looking at for kelp farms. So that's just kind of give you a little bit of context of how some of this work is being used in the news. So this is far away from me on that data pyramid, but it is interesting because it's really coming up a lot now. And a way that I really hope you all will give us some feedback is through the Alaska Ocean Acidification Network. So this QR code is a link to a survey for our 2024 discussion series. So the last time we had a discussion series, and this was from stakeholders and user groups from all around the state, was in 2022. And that series we held the same way. We survey and we ask groups to tell us what they want us to tell them. I am doing the monitoring. I'm one part of this network. I'm not the expert on all things. And it's a way to gather all the information that's most important to you and your economy and your life and your culture and the way that you eat and interact with the ocean. And tell us what you want to talk about and what more information we can give you. A lot of times we don't always know the answer, and we can go figure that out and then bring it back to you at these discussion series. So I hope you can take advantage of that if this is interesting to you. And then here is my QR code to my website, so you can go here if you want to take a look at any of the new reports that we have out, or models or results about anything. And thanks so much for your time today, and let me know if you have any questions or comments.
Thank you so much, Natalie. Thank you. Does anybody have any questions? Natalie, I have a question for you. You said if you knew two factors, you'd be able to calculate the other two. How complex is that process?
It is not complex. We have like a wide variety of tools depending on what kind of program you want to use on your computer. So we have Excel tables, we have MATLAB code, we have Python code, all that sort of stuff. So what the community has made for us, this is the global ocean acidification community. They make these software packages that you can download. It's all free. I can send you a link to one, and you can input your physical parameters, that's temperature, salinity, depth, and then you enter in your chemistry depending on what you have. And so it allows you to choose your input pairs. So if I have water samples in the lab, I'm inputting total alkalinity and dissolved inorganic carbon. It also allows you to put in the nutrients because that affects the alkalinity, so you could put in the silicate or phosphate if you have it. It's not necessary. And then you hit a button and it does the calculations for you. If I'm doing the sensor work, the sensors are measuring CO2 and pH, then I put in those, and then it calculates everything else.
Okay, thank you. Any other questions? Yeah.
I got a question. So it sounds like you're getting a lot of data on acidification and pH at different spots. How much linkage is there of that acidification data to like fish populations? I mean, I heard you mention big one was the carbon. I'm not a scientist, so what were the snails basically? Is what
the pteropods.
Pteropods, right? So I'm hearing that there's some kind of connection data-wise to the population of pteropods to acidification. Have you found any other connections as far as acidification and pH data with like fish populations or actual things like that?
Not yet. That's an area that everybody is working towards. So the number one group that is working towards that is the Kelly Lab. They're here at University of Alaska Fairbanks, and they have what I think it's called a tipping points project. So if you went to the network site, then you could go through all the different projects, or maybe even just click on that species response, like the picture of the salmon, and it'll take you to a link of the Tipping Points project. It's really good. They have a video about it and everything, and they're working on the linkages between the pteropods and the prey of pink salmon, and then all the way up through salmon. So that's an ongoing project that's going on right now. So those results will be coming out in the future. The next project that we're trying to work on is get onto the fisheries surveys in the Gulf. So in 2022, we got on a fisheries survey in the Bering Sea. So that was those little circles on that Bering Sea map where we were able to put the data into the ecosystem status report. We're working with partners right now to do the same thing in the Gulf. So this is going to take a while. It's going to take us a while to get onto these fisheries surveys so that we have the exact fishery data that they're getting with the chemistry at the same time. This is something new. Usually everyone's going out at different times because we're just really limited on resources. So this will give us the fisheries information that group is doing on population size, health, numbers, population, everything, and then be able to partner that with the chemistry. So I think that what we're going to see in the future is there's a really excellent model on the Gulf of Alaska led by Dr. Claudine Howery, and as she starts to incorporate some of the fisheries metrics into her model, I think you're going to start seeing a lot more of those Gulf species in there. Right now, the funding that I have is mostly from NOAA. So National Oceanic and Atmospheric Administration is under the Department of Commerce, right? So they're about money. So they're surveying all of the big dollar commercial fisheries. So the number one is pollock. So there's a lot of monitoring for that in the Bering Sea and the western Gulf of Alaska where the juvenile pollock are. And in the future, we're going to start working on some of these other commercial species for the Gulf. And there's going to be more and more work on the non-commercial species. So that's going to be a lot of the subsistence and personal use fisheries. So it's not there yet. Right now, they're still just ticking off the species lists. So that's all on the network too. So they all react very differently. Cod are different than pollock, or different than sole. Snow crab are different than king crab. And we don't have a lot of the species results from the fish in Alaska, especially when you're not talking about those super high commercial fisheries. So that is coming. And if you have a particular fishery you want more information on, then the discussion series is a good way to tell us that, because the feedback from the community is what will tell the agencies what to fund. So we want to study what you want. We don't want to pick what we want to study. We want you all to tell us what to study. So that discussion series is a really good way to give us that feedback.
And that was the first QR code you showed us. That's okay to share, correct?
Oh yes, and let me pull that back up here. And I can email this to you too, Christina, and you can forward it on your other newsletters.
Yes, absolutely. Thank you. Any other questions? All right. Thank you so much, Natalie, for joining us today. We really appreciate it.
Thank you for your time, everybody, and letting me join you. I hope to see you in person next time.
Yes, absolutely. Thank you.
Good job. You did a good job. Hey, you did a good job.